Hello my dear students, welcome to the MS Science Academy. Myself Meenu Srivastava, and today we are going to discuss about the microbiological assay of vitamin. As we have discussed in the amino acid assay that it is an important growth factor for the microbiology, for the microorganism, and same as the vitamin is also a special growth factor for the growth of microorganism. So in any medium, if we are just avoiding the vitamin and after some time we are adding the adding the vitamin in the different concentration, so the growth of microorganism, so the effect of growth of microorganism we will observe. So the growth or the response of microorganism is proportional to the dose concentration which we are adding. So this is the principle behind this, the microbiological assay. So let's get started. Now, principle to measure the ability of test organisms to utilize the substance being assayed under a proper nutritional condition. Okay, so here the ability of test organism to utilize the substance being assayed. The microorganism utilize those components, utilize those vitamin B12 or the other vitamins so that they can grow and they can show the response. Okay. Now, the growth of test organism is proportional to the dose or amount factor is added to the medium. Okay, so growth the growth of test organism is proportional to the dose, or we can say the amount factor, the amount of factor added to the particular medium. Means if we are adding the more in concentration of that growth factor, so the microorganism will show the more growth or more response, we can say they shows. Now, the microbiological assay of vitamin has the two assays, or we can say the two method. First is the Titomatic method, in which we can determine the microorganism growth by the titration reagent, and these titration reagent are generally it's a sodium hydroxide. In the turbidity metric method, the growth of microorganism is determined by measuring the light transmittance by using a spectrophotometer. Here the other now in this lecture we will discuss about the turbidity metric method. In this preparation, first the basal media preparation is the most important preparation. In this board you can able to see the basal medium content. So by this you are adding these content and you will prepare the basal media up to the certain volume. The pH for that is the 6.8. Now second preparation is the test microorganism. The test microorganism is selected that must be able to utilize the free cyanocobalamin. Cyanocobalamin is the vitamin B12. In this lecture I have included that the microbiological assay of vitamin B12. Okay, this is the main factor. Now, so the test organism is selected in that type that they can utilize the free cyanocobalamin. For that, Lactobacillus leishmani is the important, or we can say easily available microorganism. Now, second is the preparation. Now, the third is the preparation of inoculum. The preparation of inoculum is we can say it's a tedious task. First, the transfer the loopful of LL, that is the Lactobacillus leishmani, from recent subculture. Whatever recent subculture you prepare, that the Lactobacillus leishmani, you just take a loopful into 2 tubes, each containing 10 ml sterile medium. Now, incubate those tubes 18 to 24 hours for the 37 degrees Celsius, and after the incubation time period is over, just centrifuge them. Okay, just centrifuge them. So the centrifuge the culture and decant the supernatant. After the centrifugation, just decant that supernatant and just take the suspended culture. Okay, so the suspended culture is taken and culture into the suspension medium. In the suspension medium, I have given the following chemical content which we are taking in the suspension medium: yeast extract 0.7 gram, peptone 0.75 gram, dextrose 1 gram, potassium hydrogen phosphate KH2PO4 is 0.2 gram, tomato juice filtrate is a 10 ml, and the sorbitol monoolate solution is the 10 ml, and water is the 100 ml. This is the suspension medium content or the composition. Now, Again, after just culture into the suspension medium, incubate them, and after again centrifuge again. Okay, so just remove the supernatant, and once again the suspended cultures are, or the cells is collected, and just again it put into the 10 ml suspension medium. Now aseptically transfer. Now again, just after adding into the suspension medium, aseptically transfer 1 ml of the preparation into again 10 ml sterile suspension. Why we are doing so? We are doing so like that for removing all the other content, except we want this are just a test microorganism. So with the help of centrifugation, decantation, just we are removing the other microbial or microorganism, just we are receiving our microorganism, our test microorganism in a pure form. Okay, so after that, after doing this 2 or 3 times, just we are receiving our suspension, which is used for our inoculum. Okay, now take the, what is the reagent? For the reagent, first the standard B12 stock solution is prepared. The standard solution of the cyanocobalamin, that the vitamin B12 preparation, is prepared in the concentration of 1 microgram per ml. For that we are using the 25% ethanol. 25% ethanol we are using to prepare 1 microgram per ml concentration. Now just dilute these preparations for the concentration of 0.01 to 0.04 microgram per ml. Okay. Now the fifth requirements, that is the preparation of test solution. The test solution of cyanocobalamin is prepared, whose concentration is to be determined. Now we will see with the help of chart that what are the preparation, what are the test tubes, and how much test tubes for the standard, for the test samples we will take, and how we will proceed further. Now I have given one chart, and with this chart I will show you that how much standard vitamin B12 preparation, basal medium, and distilled water we will add in each and every test tube. For the standard preparation, there are the 10 test tubes we have to take, and for the test preparation we have to take the 4 test tubes. For the standard test tubes, just take the preparation vitamin B12 preparation is 0.5, 1, 1.5, 2, 2.5, 3, 4, 4.5 and 5 ml. In the test preparations, test preparations we will take is 1 ml, 2 ml, 3 ml, 4 ml. Means in the another 4 test tubes we will take the 1 ml, 2 ml, 3 ml, 4 ml test preparations, test sample preparations. Okay. Now in each and every test tube for 1 to 14, in all the test tubes add the 5 ml basal media, and just make up the volume is just like that, the total volume, that standard preparation or the vitamin B12 preparation plus the basal media preparation plus the distilled water preparation, when we are adding it, just combinedly it will take the 10 ml. So like that, here just add the distilled water 5 ml, 4.5, 4, 3.5, 3, 2.5, 2 and 1, 4.5 and 0, and like this 4 ml, 3 ml, 2 ml and 1 ml distilled water we will add. Okay. After this, just add the microorganism and incubate all the test tubes at 30 to 37 degrees Celsius for 16 to 24 hours. Now set the wavelength at the 640 nanometer. Now take the readings for all the 14 test tubes. Now just draw these values on the x-axis, concentration of cyanocobalamin in the microgram per ml, and here the transmittance value. Now just plotting all the values in the graph, and by taking the graph, just taking the linear graph we will receive. Now after just taking the transmittance value of the test sample, just when we are extrapolating these values on the x-axis, just we got the unknown concentration of the vitamin B12. So here now we have completed the turbidity metric method. Now we will discuss about the titometric method. Now come to the titometric method. Okay, so in the titometric method, all the preparations, either in the stock preparation, either test preparation, either the inoculum preparation, all are the same. But the difference is that just we will measure our concentration of microorganism with the help of titration. Okay, so here up to this reading, all the things you have to keep right same as in the turbidimetric method. But after this list or after this tabular form, just do one thing extra as compared to the turbidimetric method. Now what we will do, after this volume makeup, sterilize all the test tubes at 121 degrees Celsius for 15 minutes. Now cool the tubes at room temperature. When the room temperature achieve, then inoculate a drop of Lactobacillus leishmani in each and every test tube, either the standard and either the test. Now incubate these test tubes for 62 to 72 hours for 30 to 37 degrees Celsius, which is the normal temperature for the growth of microorganism. Now after incubation, all the test tubes titrate with 0.05 normal NaOH. In the lab, I think you have seen the burette preparation. I think you have seen in that all such kind of the burette you have seen. In that you will take the 0.05 normal NaOH. Okay. No, now with the in that lower side you will take the beaker. In that beaker you will take small sample of our such standard and test preparations one by one, and just as an indicator take 0.1% bromothiamol blue. Titrate these preparations with 0.05 normal NaOH until green color is achieved, For each and every preparation, when we are titrating, take the average value. What is the average value? Suppose that for first preparation, do the titration at least 3 times and take the average. Okay, so like that for the test tube number 2, do the readings for at least 3 times and take the average. Now after taking the average of the titration value, now record all the titer readings and plot a graph of titration value in 0.05 normal NaOH. Okay, against the concentration of cyanocobalamin. So now this is the concentration. I will draw with the black pen so you can easily visible it. Now concentration of vitamin B12 I have written here. Okay, and this is the titer reading. Now the linear graph is obtained, and just extrapolate your test readings. Now again, when you are just extrapolating these readings, and on the y-axis when you will meet it, you can determine the concentration of vitamin B12. Okay, so this is the concentration of unknown cyanocobalamin. Okay, so I hope you have understood the concept of turbidimetric method and the titometric method. I hope you have enjoyed my lecture. Any doubt, kindly ask in the comment section. Otherwise, please like, share and subscribe. Thank you.